Yeah, I guess, Terry. Go ahead.
Thanks. Good evening, everybody. My name is Dani Vargas. I'm very grateful to be here with you all. Thank you for taking the time to come out and to learn and to see something new. I was born here in San Antonio. I grew up in southern Missouri on a farm, ranching and farming with my family. And we moved back when I was 13 years old, and I went to high school here and then took off on this wild adventure of figuring out myself and how to be a good human being. And along the way, I learned a lot about plants and animals and living close to the land, and came back home around 2019 and spent a lot of time with my family and started gardening in the backyard. And then in 2023, I got the opportunity from the support from my mom to attend the Soil Food Web School. That's Dr. Elaine Ingham, who's an amazing soil scientist. And so I took their courses and studied all the way up to being certified for doing the microscopy work. And it really, I learned so much about all of the experiences that I'd had as a young person, working on biodynamic farms, organic farms, homesteading, planting trees. Like, it all kind of brought it into focus. And I learned a lot about why I was doing some of the things I was doing when I was farming, and the importance of the soil health to anything related to growing healthy, nutritious food and livestock as well. So I'm really happy to be here to share a little bit of this information with you and some of my experience. And I welcome questions at any time. If you need some clarity or anything, just let me know, and I really want to make this accessible, and I'll try not to dive in too deep, but also want to give you a good amount of information. And the most exciting thing is when we are going to take these samples that we just pulled, thanks to Mike, right? We took a sample of the compost over here, and we took some samples of these vegetable beds, and we're going to actually look at them under the microscope and see what we see. It's going to be an adventure. So thank you all again, and yeah, we'll go ahead and get started with a tour of the soil food web. Okay, so here's a nice diagram. Thanks to Dr. Meleka Beharan for this diagram. This shows the entire food web and how it's all related, and it breaks it out into these trophic levels, which we're going to kind of talk about. At each trophic level, you have a grouping of different organisms, and they play different roles in the food chain, in the soil food web. And I don't know if you guys remember, like, elementary biology. You might have learned about the food chain, and there's often social metaphors about that, about being the small one on the food chain, stuff like that. So this all is very relevant to that. So how it works is, of course, nothing would ever grow on this planet if it wasn't for the power of the sun. And so the sun gives us the gift of the sun's rays, and the plants take in that energy and they grow their beautiful leaves and flowers and fruits sometimes, and their roots. And they also transfer some of that energy into photosynthetics, which are oftentimes now recognized as root exudates, which we're going to talk about. It's very important to this whole system. And so that energy is transferred into both organic matter, the plant body themselves, and those exudates. And then the organisms, the bacteria, the fungi, the nematodes, the protozoas, the arthropods, the microarthropods, they all have relative reciprocal relationships. And we're going to explore that. We'll come back to this chart quite a bit during this presentation. So the main organisms I'm going to talk about today are the bacteria. They're kind of like the foundation of all life on this planet. There wouldn't really be much higher order life if it weren't for the bacteria. And we're going to talk about regular old, speck globular bacteria, single-celled organisms, as well as filamentous bacteria. There's a distinction there. We're going to talk about fungi, both the beneficials and what we consider problematic or indicator species when we're looking at soil or compost. We're going to talk about protozoa. These are, once again, still single-cell organisms, but they're a little more complex. And those are your amoebes, your flagellates, your ciliates. Then up higher in that trophic scale is your nematodes and your microarthropods. We're going to talk about all these organisms and how they're related in the soil to create nutrient cycling for those plants to be able to grow. Um, so, starting with bacteria, I'm sure you all have heard about microbiomes. We have bacteria all over us, inside of us. The surface of the world is covered in bacteria, and these guys have each bacteria has different enzymes that they use to break down certain materials that then becomes food for them. And they're the heavy lifters. They do so much catalyzing of nutrients every day, every moment. Um, so when looking at them under the microscope, they're usually clear, colorless, and they can be motile or non-motile, which means they can move around or not. And I put a little circle right here on this little black dot. That actually is a bacteria, and the reason it's black is because when we're doing this type of microscope work, we are shadowing. It's called shadowing microscopy. So normally the light would pass right through them, and you wouldn't see. They would be invisible, just like whatever water they're in. But with the shadowing, they that little black dot is their shadow, and that's how we can see them. And you see all these other little halo looking things. Some of those may be bacteria as well, but they're not in focus, and so they're not going to look like that. But when we count them, when we're doing assessments, that's what they should look like, little tiny black dots. Not all bacteria look like that. That's just a cocci bacteria, which is just a dot. There's some that are more like rod shaped. There's some that are in longer kind of chains like that. There's all different kinds, and so we um, learn about the different ones, and in order to know what we're looking at. We don't know exactly the species, but we can more or less know what their niche is as far as bacteria in their role as at that trophic level. And so the favorable conditions for bacteria, which is like right temperature, moisture, and food, they'll reproduce every 20 minutes. So these guys, and they do it by fission. They just make a copy of themselves, and then they're ready to go. And they're very adaptable. Bacteria have been found living on like asteroids and on the moon. And like if they don't find favorable conditions, they have a way of going dormant, and then they can awaken themselves just like a seed in a way when those conditions present themselves once again. Um, so we also are going to talk a little bit about some disease-causing bacteria. These are like, I don't know if you ever go to the ocean in like July or August and people talk about vibriosis. It's like a kind of a serious illness you can get from swimming in the ocean if you get infected by Vibrio bacteria. Vibrio bacteria are one of these disease-causing bacteria that are often found in only anaerobic conditions. And so when we talk about the soil food web, we talk about biologically healthy compost, we're going to talk about the difference between aerobic and anaerobic processes. And the preference for the soil food web sort of view on things of like soil health is to avoid anaerobic processes, avoid anaerobic organisms for a number of reasons. One mainly being that the aerobic organisms are highly more efficient at breaking down material anyways. And then the other reason being that anaerobic processes sometimes have secondary effects on other organisms and plants that aren't beneficial to organisms, plants, including humans, right? Because like I talked about Vibrio, we don't want to get sick from our compost. Keep it keep it aerobic. So the next group of organisms still in this second trophic level is going to be the fungi. Now, bacteria, if you guys have ever learned a little bit about compost, they talk about your browns and your greens, your N's and your C's, and the bacteria tend to prefer more nitrogen-rich foods, and they have the enzymes that allow them to break that down. They also tend to be more nitrogen than carbon in their bodies. We'll talk about that later. Now, the fungi, they're the opposite. They're more carbon, and they have the enzymes needed to break down woody materials, lignin, chitin. Say that right, chitin. So like they have those enzymes. So they specialize in those more woody substances. As people in mycology know, like that's how you inoculate logs. You don't inoculate. You inoculate straw. Not alfalfa or something. So there are. Oh, excuse me. I jumped ahead a little bit. The actinobacteria. So they used to think that actinobacteria were a type of fungi. They used to call them actinomycetes, but they realized now that they're actually bacteria. And the reason they were confused is because they will form filamentous strands just like mycelium, but they don't branch the way mycelium do, and they don't have septum the way mycelium do. However, they are really important to the food web as well. And they're these little bacteria that form a little sheath around themselves, and they form these little colonies as to protect themselves. And they actually have a different set of enzymes than the guys we just talked about, the bacterias. They can sort of break down some of those more woody materials, or begin to. And these guys are really important to have, and they're also an indicator species of being facultative anaerobes. They can exist in both aerobic and anaerobic environments. So if you are looking at a compost pile, or you're looking at soil and you see a bunch of these guys, that's an indication that maybe there's not adequate oxygen in your soil or in your compost, and you need to address that. And that's usually if it's below 6 parts per million. But once again, clear and colorless. The only reason we can see this guy right here is because of that shadowing technique, once again. Otherwise, this would be totally clear. You wouldn't know that it was there. Okay. Any questions so far? No? All right. So I started to talk about fungi, so let's continue on that. So the beneficial fungi, here's a great example of one right here. This is a saprophytic fungi. These guys, like I said, break down woody material. They tend to have a uniform diameter. So you see that? It's nice and long and filamentous. There's not any big bulges or anything. There's a little one there, but it's pretty much uniform. These guys have a lot of different colors. I've seen them brown, dark brown, purple, orange, yellow, tan, gray. Like they're just, and it's really cool. They get their color from these crystals that are formed on the outside of their mycelium, and the way that they reflect the light. It's really cool how they do that. And these guys here have a uniform septa, which, if you're familiar with mycology, you know that each mycelial cell is divided by a little cell wall. That's your septa. And so the saprophytic fungi are really important to have in your soil or in your compost. They're going to break down a lot of that organic material into humics or fulvics, which are going to be available to other fungi to use, available to other microorganisms, and make nutrients available to plants. And we'll talk about that a little bit later, how that works. I just want to touch a little bit on the mycorrhizal. I'm sure that's a big buzzword nowadays. People are really excited about what mycorrhizal can do. There's a lot of different ones. I don't want to get too deep into it, other than to point out that these guys are also really important fungi in the soil. They tend to not really be in compost unless you intentionally inoculate them, because they have symbiotic relationships with the plants, which means they have to have living plant roots in order for them to be alive. Because those plant roots, once again, take the energy from the sun, they put those exudates down, and they form a relationship with those fungi right there at the zone of the root. And they get nutrients from the plants. The mycelium get nutrients from the plants, and they transport water and other nutrients to those plants that may not be accessible to those plants if those fungi weren't there. A great example of this is phosphorus, and we see this a lot in our soils in South Texas that are particularly alkaline. You pull a soil test on from A&M, more likely than not, it's going to be deficient in phosphorus. It's not because the phosphorus isn't there; it's that it's not accessible. It's just bound up in the soil matrix with these other cations, which talk about soil chemistry at a different time. But these mycorrhizals are able to transport phosphorus directly from their mycelial networks to the plant and get them the nutrients they need directly. Any questions about that? Cool. Moving along. So some problematic fungi. And why are they problematic? Well, they have a role too, just like the actinos. They are problematic because they might cause a disease for a plant. And they're problematic because they're, once again, an indicator species of a low oxygen environment. And the umycetes, these guys right here, they tend to be colorless and clear. They're kind of lumpy. They look almost frothy and bubbly. They don't have clear septa. They're not uniform. Their diameter is not uniform, so they'll be like skinny here and wide over there. Question? I mean, they're what? Protists? I don't think so. Uh, Check it out. Yeah, I mean, I was thought that they were like kind of like I put Irish potato famine here because they're kind of in the classes of like molds and like mildews and things like that. Um, so yeah, the cause of the Irish potato famine was a type of oomycete. I'm not sure which one, don't recall, but look it up. That's what Google's for. Yeah, and check anything that I say honestly. Like they're fungus like. Fungus like. Thought of as fungus, but then they just realized they're just a little less related. They're not modified. Okay. Cool. Good to know. Hey. I'm a biologist. No, I love to learn that. So I will not include them with the fungi. What would you say that they're protists then? They're protists. Okay, just they're fungal, they're fungi-like protists. Exactly. Okay. Great. Okay, moving along. Thank you for that question. It's good. The next, yeah, we're still on the second trophic level. So moving along to protozoas. So here's an example of a protozoa. That's a testate amoeba. They're probably one of my favorite amoebas and protists that I see in my samples. They have these little shells or a test that they make themselves, and they live in there. And they kind of like have a little opening where they stick their little arms out and they eat. And when they reproduce, they'll build an empty shell first, and then they'll divide into the new shell and then separate. So under optimal conditions, every 8 hours you can have these guys reproducing. And these guys are pretty much bacteria eaters. They will eat a lot of bacteria, which we'll talk about why that's important in a little bit. Another protozoa to talk about here is the flagellate. Here's an example of one right here. They often kind of have a football shape, but not always. They can be all different kind of oblong shapes. They have flagella, which are these little cilia, usually one or two, and that's how they propel themselves through soil media. I like to think of them kind of like Indiana Jones with the whip. They like grab onto something and kind of swing, move through stuff, and you'll see them. They move pretty quick. If we see one, we'll be lucky. They kind of bumble around, and they're really kind of hard to keep track of. But they move like that, and they just go around and graze on microaggregates and bacteria and have a feast all the time. These guys are eaten by microarthropods and nematodes. And what's that? They're good. Yeah, these guys are good. You want these guys? They're a part of like they're bacteria predators, so they keep the bacteria populations in check. If you don't have these guys, then you'll have a proliferation of bacteria usually. Um, and really interesting side note is that when they're dormant, they form these really beautiful cysts, and there's all different shapes and sizes, and some of them are very beautifully adorned and look like little ice crystals and really filigree patterns and things that when you have time to just Google cysts of flagella, check it out. I mean, I nerd out about it. I don't know, you guys might. Okay. So the third one, protozoa ciliates, also an important part. They do, once again, they're one of these indicator species of anaerobic conditions. However, they're going to be there. They're perfectly fine to be there. If you have a lot of them, you'll see these guys mostly in like pond water or like streams and more. They're very much in like aqueous environments. You might see them in like really wet compost. And they are also bacterial feeders. They eat a lot of stuff, and they move with a purpose. They're just like boom, 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 and they got like all these hairs. Like that's the difference. Just one of the ways to differentiate them between the flagellates. They got a lot of hairs, and the way they move is really kind of cool. Maybe we'll see one. Any questions? Okay, so we've kind of talked over all the second trophic levels. Now maybe we're kind of in between here because we're going to talk about nematodes. And nematodes, there's a lot of different types of them, and they serve a lot of different functions. It's a really whole interesting topic just to talk about nematodes. But generally, we're going to say they're between 5 to 100 micrometers thick. They're between 0.1 and 2.5 millimeters long. They're a type of round worm, actually, single cell round worms. They have like these outer shells made out of chitin, once again, so they're very durable creatures. There's bacterial feeders that primarily eat bacteria. There's also some that are like omnivorous. They can kind of switch around. There's fungal feeding. These guys will have like a little spear in their mouths. They slither up to a fungal hypha, and they'll puncture that hypha and suck out the cytoplasm. And there's the and oh, also interesting aside, if you want to do some YouTubing later, there are fungi who will hunt fungal feeding nematodes. They have a way to capture them, and then they trap them and they eat them. So cool. So pretty wild. Then there's the root feeder. So these guys, I wouldn't be excited to see a root feeder in a sample if I saw a lot of them. If I see one or two, they're there. They will go and they have an even bigger spear in their mouth, and they'll go up and they'll puncture a plant root and they'll suck out the juice of the plant root. If you have a lot of these guys, they could be a problem. They can go inside of your roots. They can reproduce in there. Like they can cause a lot of problems for the plants. And then the last ones are the predator nematodes. These guys, I call them huge monsters because compared to the other nematodes, they are gigantic. And they eat other nematodes. And they have teeth, and they're scary. I mean, maybe not, they're like tiny, but to me, if I was a nematode, I would be afraid of the predator nematode. And they are like, it's like Jaws, it's like a big shark. Hmm. That's a great question, and there's really no way to know, I think, unless they tell you and you believe them, or you extract them and look at them for yourself. When they just say beneficial nematodes, like, I don't know what that means. Are there even really nematodes there? Question in the back. Right. Yeah. I want to try. I ought to do a YouTube video on that and test them. See what's actually in here. Question in the back. Oh, repeat the question. Yeah. Okay. Sorry about that. So the question was, what is in those nematode packages that you buy for your garden? And you heard my answer. So, any other questions that you were like, what was that question? I'll try to refresh. No. We don't know until I mean, honestly, I'm a skeptic. I've always been a skeptic. I'm like, I grew up in the Missouri Show Me state, so I'm like, show me. I want to see it. Right. Any questions about nematodes? Okay, we're gonna talk a little bit about microarthropods. These guys here can be between 0.2 millimeters and 10 millimeters in length, 0.08 to 2 millimeters wide. They're tiny. You would have a really hard time seeing them with the naked eye. They're consumers of bacteria, fungi, organic matter. Oftentimes, the acari, springtails, are common types of microarthropods, often called the shredders of dead plant material. They will take, plant fiber, cellulose, these kinds of things. They'll break it down into smaller pieces that makes it more digestible for other microorganisms. And so these guys very important role in the soil food web, as well as like some of the higher order predators. And as we go, we're going to get higher order. So we just covered the second, went into the third trophic level, and then the fourth and fifth trophic levels are predators, are species that we can see with the naked eye. So we're talking about earthworms, roly-polies, arthropods like soil mites, aphids, and then pretty much all insects, and then all the way up to birds and moles and voles and mice and all of those guys. So these guys are also, including us, we are part of this food web as well. So any questions? All right, I want to talk a little bit about the carbon to nitrogen ratios of some of these organisms, and this will become a little bit more relevant when we talk about nutrient cycling. I'm going to give the example of nitrogen, and how that cycles through these trophic cascades. So bacteria tend to have 5-to-1. That means that for, roughly speaking, for every 5 carbon that you have in that body of that organism, you have 1 nitrogen. And that's like more or less. It's just take it on as like generally speaking. Fungi, once again, generally 20 to 1, and that's a big caveat there because an older mycelial strand is going to have way more carbon than a younger mycelial strand. Um, as that mycelial strand is growing and adding onto its length, all that cytoplasm is there at the front, and they need those amino acids, those nitrogens as they're growing. Once they're established, they tend to fill out um, and put those carbon layers to protect those mycelial strands. They kind of thicken up like a skin. People 30 to 1. Who knew? 30 to 1. And what do they say about compost? That you want your compost to be 30 to 1 carbon and nitrogen? I've heard that before. Oh, isn't that funny? Oh, we're just walking bags of compost. Oh, what? So green leaves also 30 to 1. Right, yeah, right. We're walking bags of compost. Protozoa also 30 to 1. So if you ever felt attached to an amoeba, like I have, like you realize that, oh, it's because we're relatives like that. We're the same carbon to nitrogen ratio. Okay, nematodes 100 to 1. That comes from that. I was telling you about the chitin, the outer layer. So that's a lot of carbon. A lot of carbon in chitin. Brown leaves, once again, a lot of lignin, a lot of woody materials have high carbon. So wood 300 to 1. Now, why is this relevant? Well, let me introduce you to the poop loop. Um, so this is very similar to those trophic cascades that I was just talking about. You have the sun. The sun gives energy to living plants. The plants die eventually, and they give rise to these dead organic material. You have all these decomposers, your saprophytic fungi, your aerobic bacteria. These guys are breaking that material down. Nitrogen is accumulated in the bodies of the bacteria and the fungi to some extent. The fungi more so in the bacteria. Those bacteria are going to get eaten by protozoa, amoebae, flagellates, ciliates. So then all of that nitrogen goes into their bodies, and then when they poop those bacteria out, that nitrogen is released and goes right back to that plant. And it's in a more bioavailable form than it was when it was just in that bacteria. So we're going to talk a little bit about nitrates and ammonia in a little bit. Um, and so this is the same as happens with these guys. These predators eat those predators, same thing happens. And so this is how nitrogen is cycling through biology in the soil substrate. And it kind of just keeps on going and going and going all the way to us, vertebrates. When we flush our poop down the toilet, unfortunately, it takes it out of this loop, but some of us who have lived off grid might know about composting human manure. It's good stuff, and that closes that loop, so we are part of this as well. Any questions about this? Some people say that composted human manure is not healthy. Yes. Yeah, that's what I'm repeating right now. So this person's asking me. Some people say that composted human manure is not healthy. There's a lot of debate to that. For planting, sure. There's an entire book. Jenkins, is it? Writes the Human Manure Handbook. There's a lot of.
Think about if you're taking antibiotics or medication.
Yes. That's one of the things. So. Right, and so I've actually managed human manure systems on an off-grid like farmstead, and we would let our piles sit for almost 2 years, and we would turn them and let them sit and like really just fully break down as a precaution. And people were a little bit weird about. They're like, I don't know about putting that in the vegetable gardens because who knows what and pathogens and all that stuff. And really, so as a precaution, we put them just on our orchards, on our fruit trees. I think that's being a little overly cautious, mainly because going back to the distinction between aerobic and anaerobic, if you're having an aerobic human manure compost pile, you're having beneficial organisms that maybe won't be pathogenic because those pathogenic organisms really only show up in those anaerobic conditions. So that's a management question, but not everybody manages human manure well, and it takes a lot of planning and a lot of not a lot of infrastructure, but definitely labor to do it well. Yeah. The Chinese, yeah, absolutely. The Chinese have been doing it for years. Question. Joseph Jenkins. Yeah. Yeah, so free PDF online, Joseph Jenkins, the Humanure Handbook. Great, great book. If you want to go to the Tofka bookstore next year, it'll probably be on sale there as well. If you want to buy the actual physical copy, of course, Thrift Books and Amazon, they all have it. But that has a really great discussion about Humanure and a very, very detailed, scientific, in-depth. I highly recommend it for anybody who's interested in Humanure. Good? Okay. So, how does this relate to what my plant needs? That's a great question. I'm like, oh, you're talking about all these microbes. Well, what? So what? You know? And so here I want to talk about the succession, plant succession, or soil biome succession. So we'll start at the lowest level of succession, which is pretty much bare dirt, parent material. You're really only going to see bacteria there. And this is what you would consider as like very poor, not very nutritious soil. Not much is going to really grow there. Maybe some lichen, some bacteria, some moss. As you go higher as far as like the complexity or diversity in that soil food web, you're going to see a more diversity in the plant life and also the fungal and all the other life. And so as each one of these stages is marked by right here, this number FB equals 0.01. That's what's called your fungal to bacterial ratio. And that is derived in different ways depending on how you're testing it. But basically it's saying like, how much fungi do I have in relationship to bacteria? So if I have a fungal to bacterial ratio of 0.01, that means I've got 1 unit of fungi to 100 units of bacteria. And that's how it goes. So in the early successional, like weeds, forbs, grasses, it's pretty low. It's mostly what we call bacterial prominent soil profile or soil food web profile. You get up to like your mid-successional grasses, perennial grasses, prairies, late successional crops like row crops, corn, wheat, soy, oats. Ideally you'll want to have an F to B ratio of 1, which is an equal balance of fungal life or fungal biomass and bacterial biomass. And then as you go further along the stages of succession, you will have your vines and shrubs, your deciduous trees, conifer and old growth trees. And so really a lot of this has to do with how the organic matter is being recycled and the nutrients are being recycled. And I don't want to make it seem like it's just about the F to B ratio because it's not. It's a little more complicated than that. But this is kind of a good guideline for someone who's like, well, I really want to grow pear trees. So okay, and I did my soil test and my fungal to bacterial ratio was 0.75. Like, okay, great. So you want to go from here to here, somewhere between here and here. What do we need to do to get your soil to that correct ratio? Optimal. It's not really correct. It's more optimal ratio for growing those fruit trees. Versus somebody's like, I love kale. I only want to grow kale, collards, mustard greens, all the brassicas. I love brassicas. I'm not going to recommend them to go here or here or here. I'm going to be like, oh, kale right here. That's your FB ratio. Because actually, if there's a preponderance of fungi in the soil, particularly certain types of mycorrhizal fungi, they can inhibit the ability of brassicas to grow effectively. I've even read that there's some mycorrhizal fungi that will form relationships with brassica just to steal nutrients away from them. Right, that's what was thought. Was like for the longest time that they did not even form them, and I read a paper that said actually they do only for parasitic purposes. Yeah, check that out. Like here, right? So like early grasses would be like I don't know, like Bahia grass or like what's this coastal kind of like very yeah, very like low nutritional density, kind of like, and your later successional grasses are going to be like your gamma grasses, your like your bunch grasses, things that are like got a little bit more body to them, like they require more nutrients. Yeah, and transporting. Yeah, so like things like a good example of late successional would be like wild oats and basically any grass that has a pretty big seed germ. It's going to need that require those nutrients in order to produce those seed germs. Question.
The north in Dallas area to the north of Tarrant, they're trying to bring back the native grasses from that area. Or if we wanted to bring native grasses in our backyard, let's say, what would we use? Native grasses like the cyanodon from Bermuda, which is
Right.
really not that
Right.
native, but there's other grasses.
Yes, that's a great question. So the question is, if we want to restore the native grasses in our yard, what types of grasses will we use? And I don't know all the answers to that, but I can point you in the right direction to look into that, which is there's a company called Native American Seeds. Those guys have done a lot of research in local grasses, and they grow out a lot of their own seeds, and they sell a lot of really awesome mixes depending on where you are, in whether you're in the hill country or on the coast. Like they can make recommendations for the type of native grasses, and you can also do some of your own investigation, like try to find a untouched pasture that maybe hasn't been seeded with something else, and try to identify. I mean, there are thousands and thousands of species of grasses in the state alone. So another important part of that, though, if you're saying like taking what was maybe Augustine grass or something and transforming it into something native, there's a good chance you're going to need to address some of these soil food web issues in order for to create a conducive environment in the soil for those grasses to be prolific. Yeah, I would say somewhere between here, you know, FB, and that's 0.75. It's a ratio. Like, once again, I wouldn't take this as like, solid, like this is what it has to be, like any kind of dogmatic or like a prescriptive solution. There's a lot of other things. This is kind of a general, like what you're looking at ballpark ranges for these kind of organisms. Yes.
But how would you test something like that in your own backyard? How would I get to that ratio?
Right. So there's a couple different ways. One is you could contact somebody like myself who's been trained with a lane school and can take a sample of your soil, can count organisms, run some equations to figure out what the biomasses of those are, and then you'll get kind of a rough idea. The thing is, there's a huge caveat with that, which is that things in your soil are changing all the time. The conditions, the environment, the organisms, the seasonal changes, like it's such a dynamic thing. So a sample today from this spot is going to be completely different from a sample the next day or even a few hours later. There's so much variability. So you kind of get an idea of what that is, and then you can kind of begin to do the work of looking at it. The other way is there's a way to do a polylipid fatty acid, a PLFA test, which I'm not the biggest fan of. But what it does is basically takes there's biomarkers on the outsides of some of these cells, and they'll run it through a machine, and they'll do some other chemical processes, and it'll tell you, oh, we've got these kinds of species in there, and it's got about this much fungi and about this much bacteria. The problem with that is that I would say maybe 10% of those organisms have even been cataloged for their biomarkers. So it's missing a whole bunch of uncategorized organisms. So it's really like not a whole lot of specificity with that. Not to say that the actually looking at samples is that much more specific, but at least you're looking at live organisms versus organisms that have been processed through a bunch of chemicals and ran through machines and stuff. I don't know. Try both. I know also there's people who do like Haney tests and things like that. Those are good too. They'll tell you some kind of more or less what your organic matter is, and this correlates. FB ratio correlates to soil organic matter as well. So that's another thing. If you were like, well, I don't want to get that specific, just what's my soil organic matter? That's another way to do it. More questions? Yes.
It seems to me like the soil changes in response to the succession itself.
That's right.
We don't need to engineer it, so at least we do have
Correct. So the comment was, it seems like the succession it follows what's being planted there, what's growing there, and that's absolutely true. And also the comment was, we don't need to engineer our soil. Yes, and if you wanted to, there is a way to do it. But in order to for that change to actually take effect and be permanent, you're going to want to have those plants growing and going through their whole life cycle right there, and all of that organic material returning back to the soil so that that nutrient cycling can happen. And the only way that nutrient cycling is going to happen is if those organisms are there to break that material down. And generally, if you've ever lived like in like the Midwest or the Northeast, people will cut their pastures, they'll cut their hay pastures, and they say, if I don't cut my pasture, it's going to turn back into the forest. And it's true. The natural succession of things, if left untouched, pastures will start to, with adequate rainfall, right, will start to turn back into forests. Question in the back, yeah.
yeah, so the areas that I work in, which is my house and my college campus, um, we have used, well, we have been using solarization in the past to fill invasive weeds. We have really big problems with Bermuda grass and and I was wondering, like, because sometimes it's kind of impractical for us to use our garden, we don't usually have enough. Um, will that like kill a lot of the soil diversity? Is that actually going to do more harm than good for the solarizer?
Um, I would say yes and no. So, like, at the surface level, it's going to get really hot, and that soil is not going to breathe really well. So, yeah, probably not great. It's kind of a trade-off. Those organisms will tend to be wherever they can find moisture and air that they need. So they'll either go dormant, like a lot of the bacteria, if they find themselves in conditions like that, they'll just go dormant. And then once you take that solarization off, they'll come back to life. Fungi, they'll probably spore out under those conditions, especially right there at the surface. And when you take that solarization off and the moisture comes back into that area, then they'll spore out and they'll find food sources. And so it's yes and no. I wouldn't say don't solarize because you're going to hurt your soil microbiome. The soil microbiome is so resilient, and can respond to a lot of negative input very quickly. So I use solarization. I think it's really effective and important. It's, weed control, especially with invasive grasses and things like that. So I definitely wouldn't want to discourage you from using it, because there's ways that you can kickstart your soil biology as well, by applying biological amendments like compost extracts and teas and things like that to really make sure that any damage that you might be doing temporarily is not long term. They'll come back.
Like there's some plants that people will be like, yo, that's an indicator of poor soil. Like the dayflowers. I see them all the time, like everywhere. And someone told me they're like, dude, that's an indicator of poor soil. But like, are there some indicator plants like that?
Yeah, the day flowers, that one with the little yellow
Yes,
flowers.
in the middle, yeah.
So that one to me is less that it's a poor soil and more that there's a compaction layer, um, because those guys they just shoot out roots on the surface, and I see them a lot in like really heavy clay soils. So there's just not good infiltration. So sometimes plants can indicate that the soil isn't healthy, but I wouldn't say that it's a nutrition issue. It's probably more likely than not a respiration or nutrient cycling issue. And that's a perfect example, because like if you have really compacted soil, you're not going to see plants like wild carrots that put down, or dandelions even, that put down a nice thick taproot, because there's like no place for them to grow, so they're not going to be very prolific. Um, so yeah, it's soil science, soil testing, these kinds of things. It's really tricky because a lot of us have been kind of programmed to think about soil in terms of chemistry, like, oh, I just don't have the right minerals or whatever. And it's like, that can be true. Mineral balance in the soil is important depending on what you are growing, and the soil biology is also equally, if not more important in some cases, than what minerals you can take out of a chemical test that you're doing to look at nutrients. That makes sense? Okay, can we move on? All right. So once again, I just wanted to show this again, the stages of successions in a different way. Here's like, going from very low successional or early stage successional on up higher, into shrubs and then eventually to forest. Looking again, once again, at like this is going to be bacterially prominent soil, and then you'll start seeing a few more fungi. This is in the sweet spot. You have a balance, and then in your forest zones, because of all that leafy material, that woody material, is getting broken down. You'll have a preponderance of fungi in those older, those old growth forests, those later successional ecologies. And this is another of the same thing, just looking at in terms of nutrient cycling. So here with the bacterial prominence, you're going to have a lot of NO3, a lot of nitrate cycling. As you get more fungi, you're going to have NH4, which is ammonia, moving through. And this is also like what you'll tend to see as far as like as you go further in the succession, you'll see more, or you should see more of these types of higher order trophic predators. Okay, so just to conclude, a healthy soil will make nutrients more available to plants. We touched on that. And there's different ways that happens, whether it's through the poop loop or whether it's through these symbiotic relationships or whether it's the plant putting out those exudates to attract the biology that they want because they're needing a certain type of nutrient. A healthy soil will retain nutrients by increasing the organic cation exchange capacity. So what the heck does that mean? So earlier I talked about fungi and their really amazing ability to produce humic substances, fulvic acids, humic acids. These are like really long carbon chains that have a bunch of chemical exchange sites on them, and they chelate minerals in the soil and in a much more accessible way than some of your other cations might, like calcium or magnesium. Like they're going to really grab onto stuff and hold onto it, versus humic substances is way more bioavailable. And so when you have a good amount of humic material, those cation exchange sites are more accessible to plants and other organisms. A healthy soil will decompose toxins in the soil, microremediation. I know a little bit about this, and I can also say that there's people who are part of Central Texas mycology who know way more. Just know that it's real, like it's been documented. People have used fungi to remediate heavy metals and other toxins, and it works. It's a really amazing aspect of a healthy soil food web is that aspect right there. So a healthy soil will reduce and possibly eliminate the need for chemical fertilizers. So this one here is because of that poop loop, because of that nutrient cycling. We don't need to be adding a bunch of nitrate fertilizers and whatever else, potassium and whatever, potash, or you can have the nutrients that the plant needs directly through the biology. Now, that's not to say it's going to be overnight. Like, I'm just going to throw some biology down and boom, poof, no more need. No, there's a whole transitional period that needs to happen. It takes quite a bit of time to regenerate the soil health. But once it's there and the right organisms are in the right roles, serving the right functions for the plants that are there, you really don't have as much need, if at all, for chemical inputs, which is a huge cost saver for when you're looking at production scale agriculture. You can save so much money that way. And also it's beneficial to the environment, right? Because when you have all that chemical input, eventually it runs off somewhere, right? It ends up in someone's watershed and pollutes, estuaries and streams, and makes the frogs do funny things. And so reduce and possibly eliminate the need for pesticides. So this is a really interesting thing, and it needs more research. And I'm here calling on a claim by Dr. Thomas Dijkstra, which is that pests or bugs will only attack an unhealthy plant. And he's tested this out. He's got a lot of really interesting information about this, which is that if you have a super healthy plant, they're going to be resistant to pests, and the pests are actually going to go after the unhealthy plants because the plants are failing. Their cells are breaking down. They're starting to die, and the digestive systems of those pests can only process dead or dying plant material. This is Dr. Dijkstra's whole thing. And so, how do you get healthy plants? It's with healthy soil biome. So, if we're really paying attention to soil biology and we're really supporting those plants to be the healthiest they can be, then it's going to greatly reduce the pest pressure and reduce our dependence upon pesticides, fungicides, things like that. And then this is my favorite one right here: reduce water, especially living where we live and the conditions that we have of drought and not everybody having equal access to water and clean water. Being able to reduce and conserve our water inputs is super important. And by building soil structure, by having more organic material, having more fungi, um, having deeper roots, better soil infiltration, better water infiltration, we can greatly reduce our water usage, and the organic material in the soil will work like a sponge to hold that moisture in place, so that we're not just wasting water through evaporation. Any questions? No. Yes, this will be on the test. Yep. Quick, take a screenshot, take a picture. It's open book. Questions? Yeah.
So there is this like a year ago or something. I bought this packet and I started using it in my garden. Most of that stuff is fairly good, like mycorrhizal fungi and but it also contained bacillus thuringiensis bacteria, and I know that there is some debate about whether it is a good idea to be using that and like how much is too much and stuff like that, or okay in moderate amounts.
I haven't heard of that. Can you?
It's usually abbreviated as BT.
BT. Okay.
Insecticidal bacteria, and there's worry that it could have negative impact on butterflies. A lot of plants tend to incorporate the bacteria into their system.
Okay, I have heard of BT. I just only ever hear it as BT. I think they use that here actually, and I would say it's probably a good thing. And there's definitely the possibility for too much of a good thing. Just like our bodies being covered with a diversity of microbiology, if you're only spraying one thing, then it could throw off the balance of the other microbial life. So really, I think if you're gonna spray things on your plants, it's best to do it when they're really young, and it's best to spray them with biology that's the most diverse possibly, and that's like doing really good extracts or teas that are activating those microorganisms. Every plant has its own native microbiome on the surface of all of their leaves and everything, and so if we can facilitate that to be healthy, then that's what we need to be doing. Now the question, I don't know whether BT can throw that off. Maybe it's possible. It's a really interesting debate, and I definitely think the potential is there if we're over applying these things.
Into like wash. That's the only thing currently that'll stop the
Mhm. Check this out though. Speaking of vine borer beetle, there's another way around that one too. Not only talking about Dijkstra and his work about having a healthy squash plant, but vetiver grass, which maybe is more of a tropical thing, but you might be able to grow it up here. Vetiver grass. Vetiver grass. So my friend Jess was just telling me about this. They, huh? Yeah. Awesome. Yeah. He's a great friend of mine. We've known each other like 20 years. Yeah. Um, so vetiver grass will attract the vine borer beetles. I don't know why, but they go there instead of going after your squash. Yeah. So just look into it. Any other questions?
Like if anybody wants to over here, and because it's over 75 degrees, they're full of trichoderma, which is like really good for the soil.
Yeah.
People need amendments for anything.
Cool.
Yes. Okay. Yes. We can't eat them, but we can use them in the soil.
So how are we doing on time?
We're doing it's 7 now, so we have like an hour.
Awesome. So that's my presentation, y'all. Thank you so much. Thank you to Dr. Elaine Ingham, Soil Food Web School, the Microbe Library for some of the pictures and the diagrams. Thank you all for your attention. I'm going to take a little break, drink some water. I'm going to set up the microscope so we can look at these samples, and then we'll spend the rest of the time just exploring these samples. Is there a preference? Do we want to look at soil first, or do we want to look at compost? Soil first. Okay. All right, we'll do the soil first then. Great. So, thanks, y'all. Take a little break, and then we'll get it rocking again. The water out of here. Okay. So we've got our slide up here, mounted on our microscope. This is the soil sample that I took. Right now we're at 40x magnification. So this is a pretty big view of things. Right away I can see there's a strand of something. There's a couple strands over here. I'm gonna zoom. I'm gonna play with my focus a little bit here. See what else I can see. So usually what I'll do, there's a whole process for when I do an assessment, but since we're just exploring, I'm just gonna move this around until we see, there's the edge of the slide. Yep. We'll go until we see something interesting. I see like some fungal, lots of fungal stuff. I saw somebody, see that guy zooming around? Yep. Yeah, possibly a flagellate. Yeah, already went off screen. It's so we're at the lowest magnification. I can go at a higher one. So let me let's go to 10x on this fungal strand. I think it's a fungal strand. Let's see, about right here ish. I'm gonna drop us down into 10x. And then I gotta rezoom. So, yeah, so now we're at 40 sorry, we're at 100x. So there's that fungal strand I was looking at. Right here. That's probably a micro aggregate, which is basically just means a bunch of humic material with a bunch of bacteria all meshed together. I love to see these micro aggregates. That's what adds porosity to the soil, helps improve like soil respiration as well as water infiltration. It gives the soil the structure that we want, living structure. Let's look. I want to see if we can look more closely at that fungal strand. Right about there. And then let me zoom in real quick. So it's kind of hard to get the whole thing like zoomed. Yeah, it's kind of probably floating at an angle, so I can't get the whole thing zoomed. It also might not be a fungal strand. It might be like a piece of fabric because it's kind of blue. I don't see any septa. I don't see clean hard ends. So I'm gonna back out here and see what else we might see. I'm gonna open this up just a little bit. Okay. Yeah, so I've got my Abbey condenser down. I'm making shadows so we can see stuff. What is that? Oh, is that a nematode? Nope, not a nematode. Let's see. What do we want to see? I see lots of micro aggregates. That's good. There's some This over here is that's some plant matter, some fiber, plant material. Let me see if I can find something really interesting for you guys. I'm looking. Oh, there's a classic one right there. It's a short piece, but look at that one. So that looks like a fungal hypha. Um, yeah, it probably could just be the movement of the vibration of the table a little bit in the water. Yeah, I don't see a whole lot of septa there, but it could just not be very easy to see. I don't know. It's kind of interesting. The zoom is not. I don't know, there's something weird with my camera, but yeah. So that's the liquids. Everything's still settling. So that right there might be a spore, possibly.
That was the hardest thing I've ever done. Because they all kind of look the same.
Yeah, I wish. Oh man, I wish I was there on Saturday. Let's see. Here's something interesting. So right now we're at 100x, and then I'm gonna drop into. I can go to 200, which sometimes is helpful. And then I'm curious about what these strands are. These guys here, what that is. So I'm gonna drop into 400 total magnification, and try to see. Oh, yeah. So those look like some actinobacteria. You see how right about there, it's properly shadowed. That little speck is about normal size for a bacillus, or a cocci bacteria, and that strand is about the same thickness. I mean, it's a little out of focus right now. Whoa, where am I going? It's out of focus, but when I focus down on it, see it's kind of got a dark line. Same with this one. That's an indication of those actinos. So what else might we see in here? I was really hoping I'd see a nematode or something. I could go back to 40x here and try to scan. This is the garden soil. Yeah, and it's slightly diluted to 10 to 1 just because it was really dark. So not seeing it's not going to be as dense as like if it was 5 to 1. But I just wanted to be us to be able to see stuff more easily, so I diluted it. And I apologize. This focus needs to be adjusted a little bit here. So let me do that. Oh, almost had it. These things can get a little tricky. Right. There, I'm trying to match the focus of the camera to the focus of my eyepieces. So that way, as I'm cruising around here, you guys can see what I'm seeing. And usually when I pull a sample, I'll go just cruising around, looking to see for anything that's moving, like nematodes or protozoa or amoebas. And this is at a very low magnification. It's only 40 times around the whole edge. That's an interesting thing down there. What is that? There's a bunch of strands. Looks like possibly some mycelial networks there. So yeah, definitely got some fungi here. So let me try to get them into focus here. See the septa? Yeah, so the color is kind of tannish yellow. That's definitely a beneficial fungi. A nice big clump of them, huh? Better go up here. So down here is more of them. Pretty much a similar looking strand. Oh. Okay, let's see what else we can find here. Yeah, so I hear you guys. Oh, I see someone moving. So I can catch him. Nope. Okay. That. Yeah, that little round thing. Yeah, so that. That one or that one. Oh, that is probably a piece of dust on the top of my cover slip. Or yeah, that is because it's at a different plane of focus. But this guy over here, that looks like an amoeba. Right there. They're not moving, but you see there's got it's either an amoeba or an amoeba cyst. Looks like if it's a cyst, it's got two walls, there's cytoplasm inside of there. Yeah. So there was a question about microaggregates. Let me see if I can find you a really good example of a microaggregate. Yeah, here's a good example right here. So this is a microaggregate. And it's just a lot of bacterial homes in there. It's like think of it as like a metropolis for bacteria. And there's a bunch of humic material in there. It's common to see if there is a lot of protozoa present, they would be grazing all through here, eating bacteria. And that brown color is from those humic substances, the humic acids and fulvic acids that help form these aggregates. Looks like we've got quite a bit of flow going. If you look right here, that is a little piece of actinobacteria right there too. So looks like my slide's drying out because you see how everything's moving a lot. That happens. It's really warm out here, so one drop dries up fast. But that's okay. We can go for as long as we can until we don't see much anymore. But as I'm cruising around, if you guys see something and you're like, what is that? What is that? Tell me and I'll try to. This is the soil from the beds. It's kind of a mix of a bunch of different places.
So can you use this type of microscopy work to determine like an FTD ratio?
Yes, I can. And what I would do, so question is, can I use what I'm doing now to determine an FB ratio? The answer is yes, I can. I would take samples a little bit differently. You probably with a soil probe, so I'm getting a full depth. I would take, depending on the reason of my sampling. There's always, before I take any sample, I'm like, well, why am I sampling? Do I want to know why this bed is doing this and why this plant is unhealthy or why these weeds are here? There's always some kind of hypothesis, right? But I would take a sample. The first slide that I pull would be just looking at anything that moves, big, large scale predators. So I'm looking for nematodes mostly. And I'd get a count of those, right? And then the next sample I would do, same test tube, same preparation and everything. I'd pull another one, and I would kind of try to randomly go through and without looking in here, just kind of pick a spot, and whatever I see, I'd be like, oh, there's this fungal strand, there's this actinobacteria. And I'm really just looking at if I see protozoa, if I see fungi, oomycetes, actinobacteria. and I have a way of measuring them through by looking at the eyepieces. I can kind of take what I know is the width of my field of view, and I can estimate what the width of that strand is, and I know the thickness more or less, and then I can figure out what the volume of that, and then I have a conversion equation that translates the volume to mass. So I count up all those organisms, I've crunched the numbers, and then for the last piece is bacteria. I wouldn't take a sample from this because there's way too many bacteria in here for me to count. I would take it down probably to like a 500 to 1 dilution, maybe possibly 1,000. So that it's in it. When I do that, all you're gonna see is like white and little like specks, little snowflakes. And I've trained my eye on how to like identify. Well, that's a bacteria, that's a speck of soil, and I'm just gonna go in like several different places at random, and try to count for 1/4 of my viewing area. How many bacteria can I count? And I take that number down. I'll do another area, do another, get an average, right? And then based on what I know of as like the volume of a bacteria, more or less, I do another conversion equation to convert that into micrograms. I also know exactly how much is in what of my drops. So I'll measure how many micrograms is in one of these drops, or microliters really. So then I'll be able to, because 1 gram is equivalent to a milliliter, more or less, and that's how the numbers all kind of crunch out, and you get the units to match up. And that's pretty much it. So I'd run the numbers, and I'd be able to tell you, well, we had about this much fungal biomass based on the equations. We had this many bacteria present, which translated to that amount of biomass. And based on the biomass, I would get a ratio between the count of those two organisms. When I do a bacterial count, I usually divide it into quarters. And it's like I'm only going to count the bacteria that I see in this screen. And this isn't a good example because, like I said, this would mostly just be white, and I just see some dots. And I'm going up and down, focusing throughout the entire depth of the drop, going all the way up and down and counting all the bacteria. And I have my shadowing on, so I'm making sure that I'm not missing anything. So I'm shadowing in and out, making sure that those bacteria are showing up in the shadows.
Use a hemocytometer to count the cells.
You maybe could use a hemocytometer to count the cells. You would have to filter out all of that junk, not junk, but all of that organic material, and make sure that you have only what you're looking for. And usually it's based on the light that refracts off of them. So you'd need to have some like standards. So you'd have to know pretty much like how the light's going to react to certain organisms, and then as the cells are running through the hemocytometer, right? So hemocytometers will move the the liquids, right, the cellular material through them. There's different ways of detecting them. So I guess I'm thinking of like one of the ways is using the light. You know of another way that the hemocytometer uses?
I thought, like from what I learned in my biology lab, hemocytometer is it's like a microscope slide with it kind of looks like graph paper. So you're able to
Oh,
put the cells in a square, and then spectrometer is the machine that uses light to measure.
so you're saying like the little channels that divide it out? And yeah, you could. I mean, I don't know if that's more helpful. I haven't tried it. Question?
I was just thinking, I think you're thinking of a flow cytometer.
That's what I'm thinking of.
Yeah.
Yes.
So that's the hemocytometer. It has like a little grid that you put your
Yes. Thank you.
the flow cytometer,
It's a machine.
scatters.
Thank you for that. Yeah, I got those mixed up. Yeah. Yes. It's cytometer. Yes, I'm familiar. They look like little H's, right? There's like a little H, and you put it in one, and it goes like whoop, and it mates in the middle. Yeah, I haven't tried those. I mean, maybe it's cool. I don't know. I've never tried it. Cool. So we can keep exploring this. We can also look at compost. Like, what are y'all feeling? Okay. All right. Cool. So what's that? Sure. Yeah. Yeah. Let me clean off this slide, and then we can do that. I also need to. I'll need to wash this out.
This might be a good symbol because it has the mycelium and the trike.
Cool.
Okay, is that enough?
Oh yeah. So we want to do the fungi first. The block first. Okay. Yeah, since I got this pretty much ready to go.
I didn't think it's that neat. Where is that at?
Distributed through Eastern Asia, from the Himalayas to the islands of Japan. It looks like it completely gains all its sugars and nutrients from the fungi. But the Asians must be really strong. I'm just living on the natural stuff.
I'm guessing if you eat it, you compost.
Oh, yeah, maybe.
Okay. This is the mushroom block. I try to get as much of the bulky stuff out of there as possible. Oh no. Got my cover slip. Yeah, that's right. Well, best, not the best cover, but we'll work with it. Got a lot of bubbles in this one. There's a lot of chunky stuff in there, but the bubble.
Yeah.
This is the opposite.
Yeah.
This is microscopy association with the plant parasite and the fungi.
So this is the mushroom block. Oh, that's an interesting thing right there. What is that? Ah, come back.
There should be a lot of mycelium in here, right?
Right. That's a weird looking thing right there. I don't know what that is. Yeah, pretty weird, huh? Looks fungal, but I don't know what it is. Could be like a little micro fruiting body. Yeah. Okay. Kind of cool. Um, over here, there's like some big chunks. So there's a bunch of stuff. That's a piece of the mushroom block. I keep seeing all these little round things. I wonder, are those like spores? Yeah. Right. Yeah. There's another one of those fruiting bodies
Oh yeah.
on the. Whoops. Ah, some interesting stuff right there at the top. Fruiting body. This is some interesting thing over here. Look at that more. Okay. So I'm guessing those are a bunch of trichoderma spores. What do y'all think? Yeah. Saw a bunch of like greenish powdery stuff. That's interesting up here. Yeah. Okay. Oh, some spent substrate. Try to get this thing from jumping around. Yeah. Okay. Cool. How much time we have left?
So it's yeah, like 4 minutes.
All right.
But that was cool.
Should I try to just throw in a little sample here of the compost real quick? I'm curious. Okay, I'm gonna try to go real fast here. Let me see. What's that? Yeah, everyone's still alive in there. That's kind of the fun of this is we're looking at straight up live organisms. No no processing, no chemicals, nothing. Yeah, so this one's a compost. Hot compost.
forget it. I should leave it in my car. They misspelled my last name, so I just put it with glitter stickers.
And then two, then you I just saw this.
Like sometimes they won't like that doesn't count. But if you're tracking or coding the native culture library, that counts. And this she already coded it. She was like,
Well, that's good to know. I can do that. Okay, that's what I think. There's like a good chance.
yeah, well, like my like I make music 777. So like some people were like, oh, this is Tiger's off. Like yeah, for the dead. Like there's so much in the Gulf of Mexico. There's so much water to
Okay.
oh wow.
So yeah, pretty dark. So this is I only did 1 to 5 dilution instead of 1 to 10, just to be quick and to kind of get a difference. This is like so much material that I don't know that I would really use this dilution to do an assessment because it's kind of hard to see what's what. Um, but I do see some good looking things like some fungi, and I don't see um, any whole lot of problematic organisms. I actually don't see a whole lot of protozoa either. I can switch to a different mag to see you see a different picture. So yeah, very bacterially dense material. Ah, come on.
that like common for compost to be more bacterial?
Yeah, I find it to be, especially around here, because a lot of people, like commercial or large scale composters, they don't let their compost piles sit there and undisturbed. They're always turning them, or they're just moving them off the yard, because that's how they make their money. So, in order for those mycelial networks to really get going, they need to be just sitting there with the right amount of moisture, so that they can. Because once they get disturbed and broken up, like it slows down their expansion into the material. So yeah, the best kind of if you want fungal dominant or fungal prominent compost, look at Dr. Johnson's Sioux. Yeah, the Sioux reactors, and there's a lot of different variations of that I've experimented with as well that produce a lot of really great fungal life in your compost. Yeah, I see a lot of really nice looking humic materials, micro aggregates. There's some spores right there, those dark. Like that's probably a spore. That's a spore. Um, that's a piece of a pollen, or yeah, probably a piece of a pollen. I see that. You'll see some wild stuff in here too. You're like, what is that? And you gotta dig, find out where did that come from. So there's a nice piece of fungal hypha right there. Oh, yeah, let me bring it back. It's because everything's reversed, so I'm used to my controls being the other way. That's a really pretty color. I'm gonna drop us into 400. So let's see. There's that nice fungal hypha right there. Whoop! I just lost it. Sorry. Nope, this way, this way, this way. Come on. There it goes. Yeah, nice dark brown color. That's wide. That's like probably 6 to 8 micrometers wide. So that's probably basidiumites. So really, we love to see these guys. They're really good saprophytic. They do a lot of good breaking down of that material in the soil. Love to see these guys. Yep.
We have a mantra for hyphae. We say hyphae for life.
I like it. I pay for life. Check out this side of the.
But we're doing like specific mushrooms. But we just like our favorite part of we haven't even got to the costumes yet, but it's gonna be fun. I just got a new projector. Like that's the movie.
So yeah, I would say there's not a whole lot of fungi in here. The ones that I have seen look good. I bet you if that pile were to sit for a good couple months, and maybe not in the middle of the hottest part of the year, you would see more. But probably when it makes its way in the beds, I see evidence of like the spores and whatnot. So in the right conditions, you'll see. Chances are you'll see more fungi come out of that. But right now, as far as active fungal material, there's not a whole bunch, but there's not like nothing. So yeah, see that's one of those. See that star-shaped thing? It's pollen. Yeah. Yep. Yeah, you want to go back? Let me see. Let me go back. Let me go back. Yeah, that thing right there.
Is that smaller one as well?
Yep. Yep. Down there, the four-lined one, and then that thing. I don't know what that is. I don't even know. Let's look at it. 400. Let me see. I gotta line.
Like, is that a sport?
Let's see here. I've never seen anything like that. It looks like a. I don't know. It looks like a turtle shell, but I don't know what that is. Could be like part of an insect's body, like an exoskeleton or something. I have no idea. Really bizarre. Never seen anything like that. Take a picture of it. I'm at 400x. I'm at full mag. Ah, I'm gonna take a picture. Okay. Okay, just gonna make sure I save it now. Um, mystery shell. Okay. Cool. Well, I think we're up for time. So don't let me keep you here, folks.